Thank you for joining me and welcome to our presentation on exploring the genetic architecture of anorexia nervosa, where we will examine the underlying genomic factor structure of anxiety-related and anthropometric traits. Thanks to my co-authors, Zainab Yilmaz, Cindy Bulick, and Nadia McCauley for their contributions to this work. We'll start off with some background on our research question and why we might be interested in the genetic overlap between anxiety, eating disorders, and anthropometric traits. We know from twin and molecular studies that eating disorders are heritable, with heritability estimates around 50%. That is, about half of the overall risk for anorexia nervosa may be driven by genetic factors. Given the current sample sizes and genomic methodologies, there's a smaller portion of that risk, between 11 to 17% of the total risk for anorexia nervosa, for instance, that has been captured by differences in what we'll call common genetic variants. We also know that eating disorders often co-occur with anxiety-related phenotypes, and co-heritability, which represents the correlation between genetic variants that contribute to both anxiety and eating disorders, ranges from 0.27 to 0.44 among anxiety phenotypes, and that is about a quarter to half of the common genetic variation influencing risk seems to be shared between anorexia nervosa and anxiety-related phenotypes and disorders. When examining the genetic correlation between anorexia and other phenotypes, two broad classes of correlations seem to emerge. So from the Psychiatric Genomics Consortium Eating Disorders Work Group, the FREEZE-2 data, which includes about 17,000 anorexia cases, demonstrated similar levels of genetic association between both psychiatric traits, which are noted at the right-hand top of the slide, and metabolic traits, which are on the right-hand side at the bottom of the screen. And you can see that the genetic correlations are around between 0.2 to 0.3 on average for both psychiatric and metabolic traits. While looking at genetic correlations in a bivariate way can be useful, using multivariate approaches, which are being developed and becoming more popular in genetic analyses, can actually improve our understanding of genetic covariation among traits. So examining a genetic risk in a multi-trait level can increase power to detect genetic associations and improve our understanding of the nature of co-heritability. So one hypothesis, for instance, in this model, you'll see that some genetic risk, which is the blue risk, might be common to eating disorders and anxiety disorders, and some genetic risk, which is the green portion, might be common to eating disorders and more metabolic and anthropometric traits. One way to conceptualize this is in terms of larger underlying genetic factors. So genetic risk that is shared between eating disorders and anxiety disorders may actually represent a larger underlying anxiety genetic risk factor. And the same on the other side with that risk that's shared between eating disorders and metabolic and anthropometric traits. One alternative hypothesis for looking at the overlap specifically between eating and anxiety phenotypes is that there might be just one underlying latent factor that's common across all of these, or that there are no underlying latent factors. And each of these bivariate correlations is representing some relatively unique bivariate risk between them. In the current project, we aim to determine whether a metabolic and anthropometric predisposition is able to distinguish genetically anorexia from other anxiety-related disorders, and whether the hypothesized two-factor model accounts for genetic relationships among these variables. To accomplish our aims, we're using an approach called Genomic Structural Equation Modeling, or Genomic-SEM. And Genomic-SEM leverages summary statistics from existing GWAS in order to model risk, and particularly model risk that is associated with underlying genomic factors. So in order to do this, first we'll identify traits with published GWAS that are relevant to our aims. We'll compute the genetic covariance matrices among these traits and then run exploratory factor analysis. So this is a list of traits that we identified relevant to anxiety and anthropometric phenotypes. So we have the eating disorder, anorexia nervosa sample, as well as several anxiety-related phenotypes and several anthropometric or metabolic phenotypes. So this is a pictorial version of the genetic correlation matrix, which is presented in the form of a heat map. So you can see on the bottom of the slide that the darker blue and larger squares are representing correlations that are closer to 1, while the darker red and larger squares on the other side are representing correlations that are closer to -1. Those in the middle with a correlation of 0 will appear small or light in color, closer to white. So we can see here that looking at anorexia nervosa, for instance, correlations are among the anthropometric metabolic traits. So we have positive correlations with vigorous physical activity and overall levels of physical activity, and negative correlations with body mass index, fat-free mass, and body fat percentage. And also high positive correlations or moderate positive correlations between anorexia and the anxiety-related traits on that lower portion. When doing an exploratory factor analysis, we did find support for the two-factor model. And ultimately, we found support for this two-factor genetic model, which suggests that anorexia loads about equally onto these two factors, both an anxiety factor and an anthropometric factor. And interestingly, OCD seems to behave in a similar way to anorexia nervosa, but with a slightly higher loading on the anxiety factor, while PTSD, on the other hand, loads onto both factors, but loads onto the anthropometric factor with the opposite sign of anorexia and OCD. As anorexia nervosa and OCD show fairly high genetic correlation and both load onto the anthropometric and anxiety factors, perhaps there are shared elements of these traits that are responsible for this high overlap, and that the genetic correlation reflects something else like compulsivity. So as hypothesized, the anthropometric and anxiety-related genomic factors did differentiate themselves, and anorexia loaded moderately onto both. Obsessive compulsive disorder, which is closely tied to anorexia genetically, may also share overlap with the metabolic component of anorexia nervosa risk, although other studies have found that OCD does differentiate on this dimension, and so I think more follow-up and probing this specific relationship is necessary. It is possible that genetic associations between psychiatric and metabolic anthropometric traits may extend beyond eating disorders, as we found that both OCD and PTSD loaded onto this anthropometric genetic factor. So it may be worth kind of looking at other psychiatric traits as well to determine whether eating disorders or anorexia nervosa, as we know now, is unique in its association with these metabolic anthropometric traits. Some limitations of the current study are that we did have a small sample size for some traits. For instance, OCD has a relatively small sample size and still building. Anorexia is currently the only eating disorder that with available GWAS summary statistics. So coming out in the next year or so will be more data on different eating disorders as well, which is very exciting. And for future directions, we are looking for potentially a broader inclusion of psychiatric and these anthropometric traits, as this may help us to understand kind of the structure of overlap among genetic disposition for these two broader classes of traits, and to determine if factor level genetic scores may actually improve our prediction accuracy of eating disorder relative risk relative to a single trait polygenic risk score. And thank you to the PGC and UK Biobank for the summary statistics that are available to use and that we were able to use for this project.